70 years. That is how long scholars, historians, and archaeologists wrestled with one of the greatest manuscript mysteries in human history. 70 years of painstaking debates, careful translations, and heated academic arguments over a collection of ancient texts that had spent 2,000 years. Years hidden in the dark, silent caves of the Judean desert. And then, in a laboratory far removed from those caves, in the clean rooms of a university in Sweden, scientists did something nobody had ever thought to do before. They asked the parchment itself where it came from. They extracted its DNA. And what those ancient molecules said back changed everything we thought we knew. The story begins, as most world-altering discoveries do, not with a grand institution or a well-funded expedition, but with a teenager and a goat. In the winter of 1947, a 15-year-old Bedouin shepherd named Mohammed Ed-Dib was wandering the rocky cliffs above the northwest shore of the Dead Sea, near a site called Qumran. One of his goats had strayed, and while searching for it, the boy tossed a rock into a cave opening, half out of boredom, half out of instinct. He heard something shatter. Curious, he returned the next day with a companion, climbed down into the cave, and found tall clay jars, sealed and ancient, standing in the dust. Inside them were scrolls wrapped in linen. He had no idea, as he pulled the crumbling parchment into the light of a January morning, that he had just made the most significant archaeological discovery of the 20th century. Over the next 9 years, archaeologists and Bedouin excavators combed 11 caves in the same region. What they eventually recovered was staggering. Roughly 900 manuscripts and more than 25,000 individual fragments. Ancient texts written in Hebrew, Aramaic, and Greek, dating back as far as the 3rd century before the Common Era. These were not just any documents. They included the oldest surviving copies of nearly every book in the Hebrew Bible. They contained hymns, legal codes, apocalyptic visions, and detailed calendars of religious observance. For the world of biblical scholarship, it was as if someone had suddenly handed us a window directly into the ancient past, a past that lay at the very foundation of both Judaism and Christianity. But from almost the first moment of their discovery, the scrolls refused to give up their secrets easily. They were fragile beyond description. Many had partially dissolved into dust, others were fused together by centuries of heat and salt air, and had to be painstakingly separated, fiber by fiber, under microscope. And the fragments, those 25,000 pieces, presented a problem that no one had anticipated. Nobody knew which pieces belonged to which scroll. Nobody knew which cave a given fragment had come from. And in many cases, nobody could even agree on who had written these texts, where they had been written, or why they had ended up in these particular caves in the first place. For decades, the dominant theory was seductively tidy. According to this view, the scrolls were the product of a single, isolated Jewish sect known as the Essenes. The Essenes were a real historical group, mentioned by the 1st century historian Josephus and by the Roman writer Pliny the Elder. They were known to be an ascetic, monastic community that had withdrawn from mainstream Jewish society, rejecting the temple priests in Jerusalem as corrupt and invalid. Scholars believe the Essenes had settled at Qumran, built a community there, and spent their days copying religious texts in a scriptorium, a dedicated writing room, before eventually sealing their library in jars and hiding it in the caves when Roman armies advanced on the region around 70 CE. It was a clean, coherent story, and it dominated textbooks for half a century. The only problem was that the more carefully scholars looked at the actual contents of the scrolls, the harder it became to make everything fit neatly into that Essene box. The texts were wildly diverse. Some were clearly sectarian. They contained rules and beliefs that matched what was known about the Essenes. But others seemed to reflect much broader traditions. Multiple versions of the same biblical book existed in the same cave, written in different scribal hands with different words and different theological emphasis. The sheer variety of the collection started to feel less like the carefully curated library of one isolated sect and more like a broad snapshot of an entire era's religious thought. But scholars had no concrete way to prove this. All they had were the words on the page, and the words, powerful as they were, could not tell you where the parchment beneath them had been made or whose hands had stretched and cured that ancient skin. That is where the science comes in. And this is the part of the story that most people have never heard. The Dead Sea Scrolls are made of parchment, animal skin processed and dried into a writing surface. Most scholars had assumed for years that the skins used were from goats, since goats were common in the Judean desert, and goatskin parchment had been widely used throughout the ancient Near East. That assumption sat quietly at the back of academic thinking for generations, unquestioned and unchallenged. But animal skin, unlike papyrus or paper, holds something that papyrus and paper cannot. It holds DNA. Even after 2,000 years of heat, dryness, and physical deterioration, the molecular residue of those long-dead animals persists, locked into the collagen of the skin, degraded, fragmentary, but there, waiting. In 2013, a research team began what would become a 7-year interdisciplinary study that bridged the gap between biblical archaeology and molecular biology in a way that had never been attempted before. The team was led by Professor Oded Rachavi, a molecular biologist at Tel Aviv University, and Professor Noam Mizrahi, a biblical scholar at the same institution. They were joined by collaborators from Uppsala University in Sweden, Weill Cornell Medicine in New York, and the Israel Antiquities Authority, which serves as the official custodian of the scrolls and their fragments. Their method was extraordinary in its delicacy. Because the fragments are priceless and irreplaceable, researchers could not simply cut pieces off for analysis. Instead, they collected what can only be described as scroll dust, microscopic particles of skin that had naturally detached from the uninscribed backs of fragments over time. These near-invisible samples were carefully gathered by the Israel Antiquities Authority and transported to Sweden, where molecular biologist Sarit Anova traveled multiple times carrying these precious samples in authorized containers, making the journey from Tel Aviv to Uppsala in order to work inside the specialized clean rooms of Professor Matthias Jakobsson's paleogenomics laboratory. The conditions required to extract ancient DNA demand extraordinary precautions. Researchers wore full-body protective suits. The air was filtered. Every surface was sterile, because the slightest contamination, a single strand of modern human DNA, a stray environmental microbe, could corrupt the entire analysis. What they extracted from 26 scroll fragments was ancient, heavily degraded DNA. The genomes of individual sheep are nearly identical to one another, which made the analysis enormously challenging. The DNA of different species, sheep and goats for instance, is similarly close. The team had to develop entirely new statistical methods to tease apart these near-identical signatures, to distinguish which fragments came from the same animal and which came from different ones. They built mathematical models to filter out contamination, to validate their findings across multiple tests, and to establish the species identity of each sample with confidence. And then the results started coming in. The first finding overturned an assumption that had stood unchallenged for decades. The scrolls were not written on goatskin. The overwhelming majority, nearly all of the fragments sampled, turned out to be made from sheepskin. 24 of the 26 fragments tested came back as sheep. This alone was significant. It meant that decades of scholarship had been built, in part, on a false premise about the very material these texts were written on. But the skin type of the other 2 fragments was where things became genuinely explosive. 2 fragments were made from cowhide, and those 2 fragments came from the book of Jeremiah. To understand why this matters, you have to understand something about the geography and ecology of the Judean desert. Cows cannot survive there. They are not desert animals. They require large quantities of fresh water and grass, resources that simply do not exist around Qumran. The Essenes at Qumran, or whoever the inhabitants of that desert community were, could not have raised cattle. They could not have produced cowhide parchment locally, which means, with simple but devastating logic, that those 2 fragments of the book of Jeremiah could not have been written at Qumran. They had to have been brought there from somewhere else, somewhere greener, somewhere more fertile, almost certainly from a city, almost certainly from Jerusalem. This was the first concrete molecular proof that some of the Dead Sea Scrolls had not originated in the caves where they were found, or in the community living nearby. They had traveled, they had been transported, and this threw the entire Essene origin theory into serious doubt. If some of the scrolls came from elsewhere, then the Qumran caves were not simply the private library of one isolated desert community. They were, at least in part, a collection that drew on a much wider world. But the cowhide discovery was only the beginning, because when Rachavi and Mizrahi looked more closely at the content of those 2 cowhide Jeremiah fragments, they found something that made the hair stand up on the back of every biblical scholar who heard about it. The 2 fragments did not just come from the same book. They came from the same book, but they were not the same version. The text on one fragment was meaningfully different from the text on the other. Different word choices, different emphasis, a different textual tradition altogether. Today, when you walk into a synagogue in Tel Aviv, in Buenos Aires, in New York, or in Melbourne, and read from a Torah scroll, the text you are reading is essentially identical across all of them, down to the letter. Jewish scribal tradition developed over centuries into one of the most rigidly standardized text preservation systems in human history, guided by an intense theological conviction that every single character of the holy text must be reproduced exactly as received. There is no room for variation. There is no tolerance for deviation. The word is the word, everywhere, always. But at Qumran, in the same cave, you find 2 copies of the same book of the Bible. And they are not the same. They represent 2 distinct textual traditions, 2 separate understandings of what Jeremiah said and meant, and the DNA tells us that both versions were brought in from outside the desert. which means these different versions of Jeremiah were not some local aberration, some Essene eccentricity. They were circulating simultaneously out there in the broader Jewish world, in cities and communities and scribal schools scattered across ancient Judea. Multiple versions of the sacred text existed side by side in circulation during the period when the foundations of both Judaism and Christianity were being laid. Professor Mizrahi described this as evidence of what he called textual pluralism, a state of affairs in which ancient Jewish communities were at that time more focused on the interpretation and meaning of Scripture than on the precise letter-for-letter fixity of the text itself. The standardization that came later, the rigid uniformity we know today, had not yet happened. The text was still alive, still in motion, still being shaped by human minds engaging with its meaning rather than simply copying its form. This is not a minor footnote. This is a window into the actual process by which the Bible, as we know it, came to be. Before canonization, before standardization, before the great rabbinic councils of the early centuries of the Common Era made their decisions about which texts were authoritative and which were not, there was this, a world of competing versions, traveling scrolls, and communities making individual decisions about what to preserve and what to pass on. The DNA evidence also helped resolve another long-standing puzzle, one that had to do with the sheer chaotic state of the fragments themselves. When archaeologists first excavated the caves, particularly the site known as Cave 4, which yielded the largest single deposit of scroll fragments, they were confronted with a scene of almost unimaginable complexity. Thousands upon thousands of pieces, many no larger than a fingernail, stripped of context, mixed together, some without any record of exactly where they had been found. In the decades since, scholars had been laboring to reassemble these fragments to figure out which pieces had once been part of the same scroll, which fragments belonged together. It was, as the researchers themselves described it, like solving a jigsaw puzzle when you do not know how many pieces there are. You have lost the box with the picture on it, and a substantial number of the pieces are probably missing entirely. DNA offered a new approach to this puzzle. If two fragments came from the skin of the same individual sheep, or from closely related sheep in the same flock, they were far more likely to have originated from the same scroll. The genetic evidence could not tell you what the text said or what scribal hand had written it, but it could tell you whether the physical material had a shared biological origin. And in several cases, this biological fingerprinting produced exactly the confirmations and rejections that scholars had suspected but could not prove. Fragments that scholars believed belonged together turned out to share closely related DNA. Fragments whose textual relationship had seemed ambiguous turned out to come from animals with very different genetic profiles, making it unlikely they had ever been part of the same document. There was one particularly striking example that revealed just how far the forensic implications of this research could reach. Among the fragments in the collection, there was a piece that had been officially catalogued as having been found in the Qumran caves. For years it had been studied and interpreted within that context, its meaning shaped by the assumption that it originated in the same place as the thousands of other Qumran fragments. But when its DNA was analyzed, the genetic signature came back completely different from every other Qumran fragment tested. It was an outlier, a piece of parchment that did not share the biological profile of the Qumran material. The genetic evidence confirmed what some scholars had already suspected. The catalog was wrong. This fragment had not come from Qumran at all. It had been introduced into the collection, possibly through the chaotic black market sales of scroll fragments that followed the initial discoveries in the late 1940s, when Bedouin and dealers sometimes misrepresented the provenance of materials to increase their value. Similarly, an Isaiah fragment that had been published as a Qumran scroll turned out to have a genetic signature inconsistent with the other Qumran material. The DNA was, in effect, calling the bluff of the historical record, cutting through decades of mislabeling and questionable provenance to say, simply and objectively, this is not what you think it is. It did not come from where you were told it came from. For biblical archaeologists, the implications of this capability are profound. The Dead Sea Scrolls have been entangled in questions of forgery and misrepresentation almost since the moment of their discovery. In 2020, it emerged that the Museum of the Bible in Washington, D.C., which had proudly displayed a collection of Dead Sea Scroll fragments as part of its permanent exhibition, had been displaying fakes. All 16 of the fragments it held were determined to be modern forgeries, fabricated on ancient leather to deceive buyers and enrich dealers. The scandal was an embarrassment for the museum and a sobering reminder of how vulnerable the scholarly world remains to sophisticated fraud when dealing with objects this rare and this valuable. DNA analysis offers a path toward a more objective standard of authentication, one that does not rely solely on the judgment of experts who can be deceived, but on the molecular evidence that parchment carries regardless of what anyone chooses to write on it. The research also shed remarkable new light on a non-biblical text called the Songs of the Sabbath Sacrifice. This is one of the most theologically fascinating documents in the entire collection, a series of 13 liturgical poems describing the heavenly worship of angelic priests in a celestial temple. The songs are considered a key precursor to later Jewish mystical traditions, and fragments of this text have been found not only at Qumran, but also at Masada, the desert fortress, where a group of Jewish rebels made their last stand against Rome in 73 CE. The presence of the same text in two very different locations had already suggested to scholars that the songs were not exclusively Essene in origin, that they had circulated more widely through Jewish communities of the period. The DNA evidence confirmed this. The fragments of the songs of the Sabbath sacrifice found in the Qumran caves turned out to come from closely related sheep, animals that could plausibly have been part of the same flock or from the same region. 3 copies found in 2 different Qumran caves shared this genetic signature. This suggested that the text had been copied and recopied from a common source or tradition, and that it had been valued highly enough to produce multiple copies. The broader implication that the spiritual and mystical ideas embedded in the songs were not the private property of one desert sect, but were part of the living intellectual heritage of Second Temple Judaism as a whole, reshapes our understanding of how those ideas traveled and evolved. As the researchers put it, the mystical tradition underlying the songs may have continued to be transmitted through hidden channels, even after the destruction of the Second Temple in 70 CE, finding its way through the centuries into the roots of medieval Jewish mysticism. The DNA of a sheep dead for 2,000 years pointed a line from the Judean desert all the way to Kabbalah. There is something almost vertiginous about this when you sit with it. For decades, the story of the Dead Sea Scrolls was told as a story about texts, about what was written and who wrote it and what it meant. The physical material carrying those words was treated as a container, valued only insofar as it preserved the written content. What the DNA research did was flip that priority on its head. It said, the container is itself a document. The skin is itself evidence, and the biological information locked inside that skin carries a kind of truth that the words written on top of it cannot falsify. This is a transformation in the philosophy of archaeology itself, not just in our understanding of one collection of ancient manuscripts. What Richavi and Mizrahi demonstrated is that artifacts are not just objects bearing human meaning. They are biological entities carrying their own independent record of history. A piece of parchment does not lie about where it came from. It does not conform to the story you want it to tell. Its DNA is what it is, shaped by the animal it came from, the region where that animal lived, the genetic heritage of the flock it belonged to. And that objectivity, that resistance to the distortions of human interpretation, is precisely what makes it so valuable as evidence. The research has inspired a new wave of similar investigations across the field of ancient manuscript studies. Scholars working on other major parchment collections, from early medieval European manuscripts to Islamic papyri, are now asking the same question that Richavi's team asked of the Dead Sea Scrolls. Where did this skin actually come from? What does the biology tell us that the text cannot? The intersection of genomics and archaeology, once considered an unlikely partnership, has rapidly become one of the most productive frontiers in the humanities. But there is also something deeply human in this story that transcends the technical achievement. Consider what these scrolls represent at the most basic level. Someone, thousands of years ago, in a world without printing presses or digital backups or institutional archives, decided that these texts mattered enough to preserve. They prepared the skin of animals with care. They mixed their inks. They sat for hours, days, years, copying word after word in the flickering light of oil lamps, with the same focused reverence that monks would later bring to illuminated manuscripts in medieval European monasteries. And then, when the world they had known began to fall apart, when Roman legions marched through the hills and smoke rose from Jerusalem, they sealed their library in clay jars, carried it up into the cliffs, and left it in the dark. They did not know whether anyone would ever come back for it. They did not know whether their words would survive the centuries, or whether the caves would simply swallow their life's work in silence. But they hid the scrolls anyway, because the texts mattered, because the ideas inside them mattered, and because some part of them hoped, the way all human beings hope when they commit something to writing, that the future might care. The Bedouin boy who found those scrolls in 1947 could not read what was written on them. The dealers who first bought and sold the fragments did not understand what they held. Even the scholars who spent decades arguing over their content were, in a way, only seeing one layer of the story. It took a molecular biologist working 2,000 years after the scrolls were hidden to look at the parchment and hear it speak in a language older than any alphabet, the language of life itself, written not in ink, but in the double helix of ancient DNA. What the DNA said, ultimately, was this. The Dead Sea Scrolls were not the work of one isolated, eccentric sect hiding from the world at the edge of a salt sea. They were the product of a world in motion, a Jewish society engaged in active, creative, pluralistic engagement with its own sacred texts, carrying scrolls from city to desert, copying and recopying, debating and diverging, preserving and losing and preserving again. The men who wrote these scrolls were not radicals cut off from the mainstream. They were part of the mainstream. And the mainstream was far richer, far more diverse, and far more intellectually alive than we had ever given it credit for being. The Bible we hold today, fixed, standardized, authoritative, is the product of centuries of decisions about what to include and what to leave out, what to preserve and what to let go. The Dead Sea Scrolls have always represented a glimpse behind the curtain of that process. But now, with the evidence written in ancient DNA, we can see that curtain more clearly than ever before. We can see the moment before the standardization, the moment of living diversity, of competing versions, of a text still in the process of becoming what it would eventually be. The caves are still there. The cliffs above the Dead Sea still rise in the same pale ochre stone where a boy once threw a rock and heard the sound of history breaking open. And in laboratories around the world, scientists continue to ask new questions of ancient things, not with picks and brushes now, but with gene sequencers and statistical models, and the accumulated knowledge of a molecular revolution that has transformed what it means to read the past. The scrolls have not given up all their secrets, but for the first time in 2,000 years, the parchment itself is talking, and we are finally learning how to listen.